migrate through the epididymis into the vas deferens. The vas deferens, you can see it runs through the spermatic cord back into the inguinal region, then runs into the anterior pelvic or pubic region, up over the urinary bladder, and then back to the posterior bladder, where the vas deferens converges with another tubule back here. It's not very clear from this illustration. We'll have a better illustration in just a moment. Converges with the seminal vesicles. Now, the seminal vesicles are going to be where most of the seminal fluid is generated. And then you can see there's another convergence here. This is called the ejaculatory duct here. This whole structure is called the prostate. So that's the prostate there. And you have the urinary urethra converging with the ejaculatory duct, for our purposes, within the confines of the prostate. So, and of course, males are prone to all sorts of issues with the prostate. So any number of things can go wrong with maturation and development in this region. But with respect to these accessory glands, you can see the three major glands listed over here. The seminal vesicle is going to be back here. We don't see it. This is actually called the atrium of the vas deferens there. The seminal vesicle is going to be back here. The prostate gland is going to be releasing a bit of the seminal fluid. And then there's another series of glands, what's sometimes called, that sort of resides within or just above what's called the perineal wall here, the perineal membrane. And these are called Cowper's glands or bulbourethral glands. And so in general, when the whole process kicks off, then of course the spermatozoa are released into this system, the channel system from the vas deferens. The fluid from the Cowper's gland will usually be first, then the prostatic fluid, the seminal fluid, and then the spermatozoa are going to be bathed in that medium. Of course, we're looking at here is this is the process of which we do not speak. Of course, this can happen in similar process in males and females. All kind of ways now that they can actually sort of create varying percentages of reversibility with these procedures. But in general, if you're going to get a vasectomy, the assumption is you're probably not in the best position to reverse the procedure. So anatomically, we're looking at the structures. We can look at a cadaver here. So up here is the urinary bladder. This whitish region here is the prostate. And as we saw in the previous illustration, this tube here, this is all we see. That's going to be the urethra, just a portion of the urethra. In fact, if you look over here down at the bottom right of the slide, you can see the four general anatomical regions of the prostate. There is the intramural or preprostatic prostate right up here, important because there's a little valve there, a little smooth muscle valve. Then you have the prostatic urethra, which extends through the prostate. Then at the perineal region, right down here, we normally call that the membranous or intermediate prostate, and that too has a little valve in it. And then the penile or spongy urethra continues down to the navicular. In the transection, and we'll talk more about this later, but there you can see there's several compartments, but notice just for our general anatomical orientation now, notice the urethra is ventral, and the larger of the blood vessels of both arteries and veins are dorsal. And then there are three, what are sometimes called erectile chambers, two of them being much more important than the third. This is called the spongy chamber here, corpus spongiosum, that sort of lies circumferential to the urethra. And then you have the two cavernous regions here, into which arterial blood is released during arousal, but has a hard time escaping because the actual compression of the tissue tends to cut off, at least temporarily, the Hilocene arteries. But this is the primary erectile tissue that you saw in the animation. In the illustration here, what I do want you to be able to do, even in the physiology class, is identify all the important structures that you see here that are relevant to our discussion. For example, you can see the bladder there. You can see the urinary bladder. You can see the prostate there. You should be able to differentiate the prostatic urethra from the ejaculatory duct. That's the ejaculatory duct there. Here's that perineal membrane right there, so that even if you don't see the Cowper's glands, I don't necessarily see them there, they're usually going to be right there, right beside the perineal membrane, which we'll talk about in the anatomy class. And then the penile urethra distal to that. A little higher magnification here, again, the bladder, the prostate here. And what we do see more clearly this time is the distinction between the seminal gland, which is back here. This is the seminal gland back here, and this is sometimes called the ampulla or atrium of the vas deferens, and it sort of acts as a way station, a temporary storage chamber, if you will. But they're two different structures. Right, so they look very similar histologically, and certainly in this illustration. But this is the vas deferens here. This is the seminal vesicle over here. Seminal gland or vesicle. And then they will converge at the prostate to form the ejaculatory duct, which in turn meets up with the prostatic urethra. So here's the accessory gland number 1. Here's accessory gland number 2, the prostate. And then do they show us the little Cowper's gland down there just above the perineal membrane. Here we have a better illustration. They can see the vas deferens here and the seminal vesicle here. This white layer up here we'll talk about later, that's the peritoneum. So that if you were to stick your head into your patient's abdominal cavity and look south, you wouldn't see any of this because it would all be covered by that peritoneal membrane. And then that sort of pinkish or peach colored tissue, of course, is the bladder. So we're looking at all of this from behind. And this grape like about the size of a walnut, more than a grape, is the prostate. You can see extensively vascularized. We'll talk about the blood vessels in this region later. And then down here are the mobile urethral glands. In the animation, there was discussion of the seminal fluid. Seminal fluid and semen technically, at least, are used interchangeably. Sometimes you'll see something of a scholastic distinction between the two, but I think that was discarded decades ago. Most of the semen arises from the seminal vesicles. The animation said about 60%. Here it says about 70%. I would say some sources you'll see as much as 75%. Point being, most of the semen arises there, and this is primarily a nutrient solution. There's a little table that we got, and if you have this table in this slide, there's a discussion later. But most of the seminal fluid is a high carbohydrate fluid that's intended to provide energy for the travelers. There's also some degree of immunological fluid in there, immunoprotective fluid, also a slightly basic pH is elevated by bases and buffers of various types. And by the way, down here it refers to sperm per milliliter, so don't confuse the rate at which the sperm is produced in a day with the sperm count. So here we're talking about 100 million sperm per milliliter. So this is sort of a density or relative abundance assessment. We'll talk more about arousal later, but you should know, and all this is talking about in the graph here, in the figure, and in the brief discursive points, is that arousal, male arousal and female too, is largely parasympathetic, involuntary and parasympathetic, whereas emission and ejaculation are sympathetic. And this will sometimes be an issue not only for your normal patients, but for your spinal patients who may or may not have impairments of these autonomic pathways. And of course, we've talked about the fact that a lot of the medications, the treatments that are advertised so abundantly on television and sports programs are designed to trigger those arginine pathways that end with the production of increased levels of nitric oxide, smooth muscle relaxation, and engorgement, generally, but especially in this region.
The organs of the male reproductive system include the testes, seminiferous tubules, epididymis, ducts, urethra, and penis. The seminal vesicle and prostate and mobile urethral glands produce nourishing fluid for the sperm. The testes subdivide into lobules that contain seminiferous tubules that produce sperm. Sperm moves from the seminiferous tubules to the efferent tubules to the epididymis where they mature. The end of the epididymis forms the ductus deferens or vas deferens. This structure ascends the posterior border of the testes, penetrates the inguinal canal, enters the pelvic cavity, and loops over and around the urinary bladder. The seminal vesicles are glands at the base of the bladder that produce seminal fluid. Together with the ductus deferens, they form the ejaculatory ducts. During ejaculation, these ducts eject sperm into the male urethra, the common passageway for urine and sperm. The prostate gland surrounds the prostatic urethra just below the bladder. The fluid it secretes goes through small ducts into the prostatic urethra. The bulbal urethral glands produce alkaline mucus secretions that empty into the urethra. The urethra passes through the penis carrying semen, a mixture of sperm and secretions of the glands. During ejaculation, the smooth muscle sphincter at the base of the bladder closes off to ensure that no urine is expelled and that semen does not.
So be sure you understand this. We talked about the preprostatic urethra. What we're looking at here is that sort of check valve to prevent the effluent from going back into the bladder. So we indicated too the seminal fluid is mostly nutritious, nutritive. The component maybe 5 to 10%, even less, from the prostate. Also elevates the pH. It also includes proteases, the function of which is not, in some cases, is not entirely clear. The prostatic fluid also releases zinc, which is thought to protect the DNA. Sometimes if there's a deficiency of zinc released in the prostatic fluid, that can lead to impaired fertility, for example.
Enter the urinary bladder. Sperm moves from the seminiferous tubules to the epididymis, to the ductus deferens, to the ejaculatory duct, to the outside of the body by the urethra.
And I think he talked about what the Cowper's gland. Remember, the Cowper's gland is primarily lubricating fluid that precedes the other fluids to ensure, for example, that there's elimination of any residual urine and so on. All right, so let's talk about probably the most taxing, to the extent that there is any taxing aspect of this discussion, the endocrine regulation of spermatogenesis. We're going to learn later, in fact, that testosterone production in the male is largely mediated by luteinizing hormone. In fact, as we indicated before, that was important for spermatogenesis. Fetally, human chorionic gonadotropin and luteinizing hormone bind to similar receptors. So in other words, as LH is being cranked up, because LH is, in order for LH levels to achieve their normal adult concentrations, that testes must undergo maturation and development. So while all that's happening, human chorionic gonadotropin from the placenta sort of acts as a surrogate or substitute for the oral LH and ensures that adequate levels of testosterone are produced in the fetus. We've ended, and that's really what this is all about. I want to make sure you appreciate that the levels of testosterone and DHT depend upon normal development of those testes, and both of these are essential for primary sexual characteristics and those secondary sexual characteristics that we talked about, characteristic of the male dimorphism, the male specific pattern of hair, musculature, changes that occur at puberty, thickening of the skin, and so on, libido. And you can see the typical age range for onset of male puberty, highly variable, anywhere from 10 to 12 to concluding at about 16 to 18. By the way, another term with which you should be familiar that can sometimes be vexing is the term virilization. So virilization would be normal in males, probably less so, almost always less so in females. And by virilization, we're talking about the developmental acquisition of male-type secondary sexual characteristics, male body hair patterns, and so on. So virilization should be appearing in the male, normal male puberty during puberty. But in females, again, we talked about what type of condition might give rise to virilization in the developing female. CAH, yeah, congenital adrenal hyperplasia. Remember what happened there? What's the overproduction of what in the developing? But from where? Correct, from the adrenal cortex, yeah. So technically what you get is hyperproduction of those weak androgens, DHEA, androstenedione, and so on. But there's so much of them that they get peripherally converted into the strong androgens that can have the sometimes unfortunate consequence of virilization. As I've indicated, not often, but sometimes these genetically female children are initially viewed as males because they have rudimentary male genitalia and so on. Again, the characteristics that we would associate with the male phenotype. But I just want you to be familiar with this term because it's really used in different ways depending upon the sex of the child. In one case, it's normal. In the other case, it may be pathological. So we've talked about these two gonadotropins. Again, remember, GnRH drives release of pituitary LH and FSH. LH, as I've already indicated, is the primary steroidogenic hormone necessary for production and release of testosterone. And FSH is going to be very important for especially ensuring that the Sertoli cell does what it's supposed to do. Remember, the Sertoli cell is important for creating the appropriate environment for the developing spermatocyte. So they have two very different roles. We've talked about the pulsatile release of GnRH. It seems to be very important, especially with the onset of the female reproductive cycle we'll talk about next time. And both the hypothalamic hormones and the pituitary hormones are going to be regulated by several gonadal hormones. Now, when you think of the gonadal hormones in the male, what comes to mind? What is it? Testosterone and DHT. All right. But you should know, and we'll talk about it in just a moment, there are also several peptides released from the testes that act to either promote or inhibit release of pituitary hormones. And my guess is that by looking at the name, you can guess what they do. Activin tends to increase FSH production. Inhibit does just the opposite. And of course, do not forget the traditional feedback pathway. So as testosterone levels go up, what's going to happen to the gonadotropin levels? As testosterone levels go up from the gonads, what's going to happen to LH and FSH? They're going to go down. Right? George, you have a question?
I thought that LH hormone was testosterone and FSH was estrogen.
I'm sorry.
I thought that LH caused production of testosterone and FSH.
That's generally true, yeah.
Estrogen.
And that's what we said. What are you looking?
The bottom line, so they both reduce testosterone production.
Oh, yes, yes. I would agree with you that LH is much more important. But what we're going to find is secretions from Sertoli cells can also regulate the Leydig cells. We'll look at that in just a moment. They're paracrine and endocrine signaling pathways as well. But yes, I would agree with you, John. So we've talked a little bit about the general function of these. Let's look at each of these hormones in a little more detail. FSH, as we've indicated, for the most part, binds to receptors on their Sertoli cells. And we've indicated the Sertoli cells are the mothering cells, the sustaining cells. And when FSH binds to receptors on the Sertoli cells will release a number of chemicals, a number of agents, chiefly paracrine agents, that have a variety of functions. So let's just make sure we understand this general relationship. As anterior pituitary FSH levels go up, the FSH will bind to receptors on the Sertoli cell, causing the Sertoli cell to release a number of things. I have a list of them coming up in just a moment, but this is probably the most important: ADP, androgen binding protein. The bottom of this slide, for our purposes, this is a list of the important products released from the Sertoli cell when the FSH binds FSH receptors on the Sertoli cell. Of course, what we're looking at here is the Sertoli cell. We can see the developing spermatocytes and eventually spermatogenesis culminating in the spermatid there. So we're going to come back to this in just a moment, but make sure you appreciate as well the spermatocyte is foreign, alien. It's an invader in the mother's body. How could we make that argument? What? Yeah, its DNA is different in ways that we haven't even begun to talk about. But these gametes are quite alien. So this immunological protection afforded by the Sertoli cells is necessary, and by the way, that sometimes breaks down and create problems. But we call that the blood-testis barrier, and the blood-testis barrier is pretty much created and generated by the Sertoli cell. It happens in a variety of ways. It actually can result in destruction of certain lymphocytes, for example. But let's turn our attention back to these products of the Sertoli cell. There's a production of plasminogen activator. As far as I know, it hasn't been quite established. This is an inflammatory cytokine or pre-cytokine that mediates inflammation. Somebody explain this to me. As FSH binds Sertoli cell receptors, this gonadal peptide called inhibin is released. And what does inhibin do? Inhibits FSH. So is it clear that this is a regulatory pathway? As FSH levels go up, inhibin is released from the gonads. And what does inhibin do? Inhibin FSH. Right? So we talked about this type of regulatory pathway in the release of growth hormone. Notice the bottom one here. As FSH levels increase, aromatase activity increases. What does aromatase do? Converts testosterone to estrogen. So estrogen production goes up with binding of FSH. But this is the most important here, reserving the most important for the final here, the ABP, androgen binding protein. Because the general principle you need to appreciate is that these spermatocytes, we can see that they're completely engulfed by the Sertoli cells, but they require androgens, especially testosterone. How can I say this? They need to be in the proximity of high concentrations of testosterone in order to develop properly. Testosterone is necessary for development of the spermatocytes in the course of spermatogenesis. However, these spermatocytes do not have receptors for testosterone. They do not have androgen receptors. So they need the testosterone, but they don't have receptors. So Can you solve this problem? How does nature solve this problem? But what do you think ABP does? What does the name suggest to you? Remember when we talked about steroid hormones? What do steroid hormones require in the bloodstream to get around? Yeah, they require a protein chaperone. So what will happen is that FSH binds the receptors on the Sertoli cell. Androgen binding protein levels go up. Androgen binding protein does a couple of things. The most important thing that it does is it binds testosterone in the lumen of the seminiferous tubule, thereby dramatically increasing the concentration. Otherwise, the testosterone would be relatively insoluble in the watery milieu, the watery lumen of the seminiferous tubule. So ABP dramatically increases the concentration of testosterone in the tubule lumen. And also, if we were actually do a sort of cytological analysis, we would see that the concentration of testosterone in these little interstitial spaces also is increased. And so really what's happening is that the level of androgen is increasing in the area around the spermatocyte by virtue of the activity of ABP. Yeah, we'll come back to that one. The other thing that happens, the other thing you can think about, testosterone is a steroid. So where are the steroid receptors located? Inside the cell. As you look at this picture, where would you expect steroid receptors to be? Are there steroid receptors on the spermatocytes? No. But in the way that I think of this, because this can get a little more complex than we need to get into it here. If we did a histological stain for androgen receptors, their Sertoli cell would stain quite heavily. So you should understand that there are androgen receptors within their Sertoli cell. Now, what they're doing there is not necessarily the same thing as they're doing out in the lumen, but I do want you to understand just the general premise here, to sort of understand our principal argument. Spermatocytes require testosterone. They lack receptors for testosterone. When FSH binds FSH receptors on the Sertoli cell releases ABP, which increases the concentration of testosterone in the proximity of the spermatocytes. And then third, probably acting to regulate a variety of intracellular pathways, the Sertoli cell does have androgen receptors, and as the concentration of androgen is increased in ways we'll talk about in just a moment, the Sertoli cell probably changes its physiology to accommodate the increase in androgen. So, but really the ABP is just a chaperone, a carrier, increasing the concentration of the testosterone in the proximity of the spermatocyte. So, I guess that's really what I've said in this slide. The only other thing I would point out is that structurally, ABP, as far as I know, may be identical to this molecule, SHBG. So H, you probably know what that stands for. Hormone. B is binding. G, globulin. S, X, yeah. Most classes say steroid because they're polite. Not this group. No, it's just sex hormone binding globulin. You're exactly right. But my point is, it's probably the same thing. ABP and SHBG, which we'll talk about in females, they're probably structurally pretty much the same thing. If not, they're very, very similar. So we've talked about, George was talking about the function of the Leydig cells. Now, we're looking at the seminiferous tubule. So this thing here would be a seminiferous tubule. This is a fixed specimen, so it's not going to be very, very realistic. But outside, beyond that seminiferous tubule, which we know we can see the light-colored Sertoli cells in there. We can see the spermatocytes. But extraneous, outside that seminiferous tubule, there are some other cells called Leydig cells that are going to play an important role in our story. So, whereas Sertoli cells are called mother cells or sustentacular cells, Leydig cells are called interstitial cells because they're sort of outside the perimeter of the seminiferous tubule, and oftentimes they're in the company of an extensive network of blood vessels. So these Leydig cells, in response to binding of luteinizing hormone from the anterior pituitary, are responsible for releasing testosterone. So the Leydig cells are responsible for releasing the testosterone. Seems to me we might have a problem here. We know that these spermatocytes require testosterone, that they don't have receptors for testosterone. And to make things even more complicated, the cell that does release the testosterone is way out here outside the seminiferous tubule. Anybody have a solution to this problem? How do we get the secretory product of this in here where it's needed to bind to ABP released by the Sertoli cells? What do we know about steroids? Are they water soluble, fat soluble? So are they going to be particularly concerned about the membrane serum, or they can just dissolve right through? And that's in fact what happens. So as the concentration interstitially increases, the concentration in the seminiferous tubule also increases. Yeah.
It says that the LH hits receptors on the Leydig cell that initiates a CAMP pathway. Is that the same as the PKA pathway?
Yeah, we use all those. So cyclic AMP again is just the second messenger for the PKA pathway. You're exactly right. And when those receptors bind the LH again, that's going to result in elevated production of testosterone, and then the whole sequence of events we just talked about inside the seminiferous tubule. Notice the Leydig cell does not have, and this is what George was talking about, this Leydig cell does not have FSH receptors, but the products of Sertoli cell secretion can regulate Leydig cell activity. So I think I've already made most of my points. I just want to make darn sure that we all understand the relationship of these various cells and their products. The Sertoli cell does not produce or release testosterone. What cell does? The Leydig cell. But again, does have receptors for testosterone. They have to be very careful there because we only partially understand what those androgen receptors are doing. By the way, the proper term for these testosterone receptors is androgen receptor because the androgen receptor binds both testosterone and DHT, and in some cases other steroids as well. And then the Leydig cell does not produce or release estrogen. And this is important. This is the point that I was making earlier that folks sometimes refuse to accept. And that's why sometimes included on tests. Make sure you understand that the Leydig cell does have estrogen receptors, and in fact, virtually every component of the male reproductive tract has estrogen receptors. Not to mention the male brain that we talked about before, and this gets very confusing. And then we talked about the relationship between the testosterone released from the Leydig cells passing into the seminiferous tubule binding to ABP released from Sertoli cells in response to what causes release of ABP from the Sertoli cell? FSH. Yeah. So just what I would encourage you to do is maybe write down all of these, write this out in your own words so you understand the various relationships. It says here testosterone is also a precursor for estrogen in the Sertoli cell. Somebody explain that to me. How is that possible? How is testosterone? Aromatase reaction. So again, the plot thickens fast. So when we talk about a precursor for estrogen, it's that aromatization of testosterone that occurs in a number of locations in the body, but especially in the seminiferous tubule. All right, so this is simply the recapitulation of the importance of these estrogen receptors. They're critical at every cells, the cells lining the entire reproductive tract, the prostate, all of these have estrogen receptors. We know that in the absence of those receptors in any of those tissues, there's going to be some degree of impaired fertility. Indeed, if there's inadequate production of aromatase, at that end, we talked about the congenital adrenal hyperplasia resulting from an enzyme deficiency. Likewise, if aromatase, you might want to think about what would happen if a child were born without sufficient aromatase. What would be the reproductive consequences in terms of the differentiation of the individual? And so again, men who lack the ability to produce estrogen or lack estrogen receptors not only have impaired fertility, impaired development, but notice it even affects the growth, what we're going to talk about from what you'll talk about in your pediatrics course, is the effect of these various hormones upon elongation of long bones. Sometimes the relationship is fairly clear, sometimes it's not. And remember our discussion of the finger length? It's testosterone that's having an effect on the relative finger length of the index finger and the index finger and the ring finger developmentally. Likewise, individuals with elevated testosterone developmentally, males anyway, tend to have a rapid growth spurt early in their childhood, and then the growth tends to stop prematurely. So they grow fast initially, and then they stop. So typically most poly Y males, something that used to be called super males, XYY or XYY, tend to be a little shorter. High testosterone males developmentally tend to be a little shorter. But notice what it's saying here is that estrogen receptors and estrogen levels can also affect growth, but in a somewhat odder way. Klinefelter's is XXY. Remember Klinefelter's? Would you expect such individuals to be taller than normal or shorter than normal? You might expect them actually a little taller than normal, right? This is physiology. Everything is backwards. XXY tends to be, I think I talked about the Ryan Reynolds movie where do you know what movie I'm talking about? He's married, and then some woman shows up to his door with a baby and says it's his. And so that creates problems with his wife, and she leaves. And then, I remember this movie better than I do. And then, come to find out, he has Klinefelter's. And what do we know about Klinefelter's individuals? Not only are they taller than normal, they're also infertile. So he couldn't have had the baby. I don't forget the whole plot now, but I just remember that there was a reconciliation at the end. Everybody forgave everybody else. But just be aware that sex hormone levels developmentally modify growth, and we'll talk more about that. It's very complicated, as I've said. The LH and FSH are both peptides. You need to be aware of, of course, that we just talked about the fact that they utilize Bob was talking about the PKA pathway. They, in many cases, will use the PKA pathway to culminate in CREB. What was CREB? And what was this? Yes, so actually it's a pathway by which peptide hormones can have effects on transcription. Yeah, we already talked about that. So with respect to the hypothalamic hormones, just be aware that deficiency of hypothalamic hormone can result in usually hypogonadism. What do I mean by hypogonadism? Hypogonadism. Well, that would be the product of the hypogonadism. The hypogonadism, the term hypogonadism, hypogonadism just implies that the gonads did not develop. And so as a consequence, you would expect, for example, a deficiency of hormone, and if there is a deficiency of, for example, GnRH, those individuals are likely to be infertile and perhaps have other issues as well. That's called Kallman syndrome. Kallman syndrome is due to a deficiency of GnRH. Notice this term. This is in your clinical literature. You're going to see this quite a bit. Prepubertal hypopituitarism. Prepubertal hypopituitarism. What does that mean? means a deficiency in the levels of anterior pituitary hormones prior to puberty, so it's going to affect their development, their childhood development. And so think about all those. If it's hypopituitarism, it can affect growth hormone likely, it can affect prolactin levels, it can affect all of those anterior pituitary hormones, including LH and FSH. And that too generally results in some degree of impaired fertility.
Isn't GnRH from the hypothalamus?
Yep, that's what we said. Kallmann syndrome is hypo.
But it says the hypopituitarism causes diminished or absent release of GnRH.
How would that be? The short loop feedback. Remember the short loop feedback? So if you had impaired feedback, you could have elevated levels, but they wouldn't do any good. It wouldn't result in production of LH or FSH. But in hypopituitarism, it just means that there is a deficiency of anterior pituitary hormones such as LH and FSH, but that in turn may result from a deficiency of GnRH. Kallmann syndrome specifically is the hypogonadism that results from GnRH. Probably the best way to say this is altered levels of GnRH through impaired feedback. So we've talked about these critical hormones that are present. We talked about testosterone and estrogens. Talked about the importance of androgen receptors, estrogen receptors. But in addition, be familiar with these three gonadal peptides. Focus, remember what I said. These are gonadal, these are released from the testes. These are peptides, these are not steroids. And their function, as the names suggest, are to mediate or modify the release of FSH. Inhibits FSH. Activin, released from the gonad, increases levels of FSH. And my guess is you can probably figure out what polystatin does. Polystatin. But the statin, so is it going to increase or decrease levels of FSH? Decrease. If it's a statin, it always is inhibitory. Anybody familiar, any music lovers familiar with castrati? What were the castrati? Right, he made a gesture. I mean, it was just a horrifying gesture. I won't even talk about it. But yeah, the castrati were, you've heard of boys' choirs, but you get a boys' choir. Suppose the boys' choir just kept being boys right up into their 40s and 50s. How could you achieve that? You can achieve that, as was suggested there, by the sort of eliminating the testes. Cake, what was it called? What do they do with cows? Or not cows? Cows don't have to worry about this problem, but the bulls, what is it called? Cake on it? Not many farmers in here, I gather. But in any case, the castrati were said to have the power of the male voice with the range of the female, but they were typically what they call counter tenors, very high range, even higher in some cases than a soprano. But what I wanted you to think about, so what would you expect in terms of the endocrine relationships of the castrati? Let's say, by the way, there was some stories suggesting this continued in Eastern Europe up until the early part of the 20th century. But what would you expect for the hypothalamic pituitary axis of the castrati? What would you expect there? How would their hormone levels be abnormal? Let's begin with the easy part. Would the testosterone levels be normal? No, they'd be diminished. What about the LH and FSH levels? Elevated. Why?
There's no negative feedback.
Right, there'd be diminished feedback. So we're going to see an analogous process that occurs in females as well. So we've talked about, I'm not too interested that you need to be concerned about the structure, but these are peptides, and just note where they're released. Activin promotes release of FSH. Polystatin tends to neutralize activin, and thereby tends to lower FSH levels. So I have a couple of slides here, we won't take time to go over them, that just sort of summarize this hypothalamic pituitary gonadal axis. We talked about the, what are these cells? What's the other term for these cells? Lighting cells and the Sertoli cells, and make sure you understand the axis and the feedback relationships between these. We have indicated that in males, testes are responsible for androgen production, and females, the adrenal cortex is largely responsible for androgen production. In the normal male, 90–95% of circulating testosterone arises from the gonads. We talked about the activity of 5-alpha reductase, important for converting testosterone to DHT. You often see that DHT is more potent than testosterone. Really, from a pharmacological perspective, potency just means what? Remember what we mean by potency in this context? It just binds the receptor. It binds the androgen receptor with greater avidity than does testosterone. But it too, this DHT is important, especially for dimorphic differentiation. For example, the reason that I am bald and most of the guys up here are not is because most of you other fellows have higher concentrations of estrogen than I have. But I have lots of DHT, which is shortening my life and increasing the propensity for cardiovascular disease. But also gives me this beautiful crown, right? I don't have to worry about combing hair every day. Male pattern baldness is largely a consequence of DHT levels development. And you can see what happens as we age. It doesn't even matter if I have more to begin with, because by the time you get to, very close to where some of you are, it all goes pop. Okay? All of you. That's why, again, you need to heed the billboard up there, and all of us need to go get testosterone injections. As far as I understand, the billboard doesn't matter if you're a man or a woman, just go get testosterone. You'll feel better for it. So, we've talked about the importance of these androgen-binding proteins. Remember, SHBG is sex hormone binding globulin. So normally, testosterone and DHT are going to be bound in transit to either SHBG or to some other blood-borne protein. Albumin is the most abundant protein in the blood, so it acts as a sponge and soaks up a lot of other things. Only about 10 to 15% remains unbound in the blood. But remember, that's the active portion. That's the active androgen. And then just a summary or just a recapitulation of what we talked about. Testosterone can be metabolized to DHT or estrogen. In the one case, in the first case, by 5-alpha reductase, in the second case by aromatase. Therefore, when testosterone is unbound and circulating through the blood, it can enter the cell to bind to receptors either as testosterone, DHT, or one of the estrogens. And when it does that, just remember the primary function is to act as a transcription factor, because it is a steroid after all, and that's how it makes a living. All right, we'll stop there.